Hello, my name is James Ropa, and I would like to share a quick summary of a recent work out of the Broxmire lab entitled Human Hematopoietic Stem Progenitor and Immune Cells Respond Ex Vivo to SARS-CoV-2 Spike Protein. SARS-CoV-2 is the coronavirus that causes COVID-19, the disease at the center of one of the worst public health crises the world has faced in over a century. And while SARS-CoV-2 is typically thought of as primarily a respiratory infection, mounting evidence has made it clear that COVID-19 is more systemic in nature. In fact, there are many non-respiratory associated complications that arise in COVID-19, and some of the most prevalent of these are hematologic complications. This is including, but is not limited to, lymphopenia, thrombocytopenia, and cytokine release syndrome. So we sought to determine whether the hematopoietic system is susceptible to being impacted by exposure to SARS-CoV-2. Indeed, we found that rigorously defined human hematopoietic stem and progenitor cells isolated from umbilical cord blood, as well as fully mature immune cells isolated from peripheral blood, contain subpopulations of cells that express cell surface ACE2. Now, this is the receptor to which SARS-CoV-2 binds to facilitate host cell infection. Hematopoietic stem cells have the highest subpopulation of ACE2 positive cells, and this suggests that these cells may be the most susceptible to infection by SARS-CoV-2, or impact by exposure to SARS-CoV-2. To test the impact of SARS-CoV-2 exposure on hematopoietic cells, we utilize recombinant SARS-CoV-2 spike protein, and this is the viral factor that binds to host cell ACE2 to drive entry to the host cell. We found that hematopoietic stem and progenitor cells exhibit decreased expansion and decreased colony forming capacity in the presence of spike protein, suggesting that spike protein exposure inhibits normal growth and function of stem and progenitor cells ex vivo. Interestingly, these effects can at least be partially neutralized by treatment with a SARS-CoV-2 targeting antibody, by incubation with soluble ACE2 protein, or by treatment with the angiotensin 1,7 peptide. When incubated with the spike protein, peripheral blood monocytes exhibit a distinct change in size and granularity. This is possibly consistent with a barren cell morphology or death. It is important to note that these cellular responses are induced by spike protein alone, which is not sufficient by itself to infect cells. Thus, it is possible that hematopoietic cells may be impacted by SARS-CoV-2 exposure even absent an actual infection. These impacts on primitive and mature hematopoietic cells may account for the hematologic complications observed in COVID-19 patients, underscoring the need to treat all facets of this disease, and this may also have important implications for hematopoietic stem cell transplantation in the COVID-19 era. Thank you.